może... Hello everyone, my name is Mikołaj Kuska and I have a great pleasure to be the chair of the first keynote lecture during our conference LAS 2024. Before I announce our speaker, the one technical note: as every year, one of the plenary lectures is an open event. So during the lecture, you can ask the questions via chat on YouTube, Facebook, or via our conference platform, and our team will collect them all for the final Q&A session. So, does life need water? This is the question asked by Dr. Janusz Pętkowski in today's lecture, an astrobiologist that has spent many years at the Department of Earth, Atmospheric and Planetary Sciences at the Massachusetts Institute of Technology, and is currently affiliated with the Faculty of Environment Engineering at the Wrocław University of Science and Technology. Janusz is also Deputy PI of the MIT-led Morning Star Mission to Venus initiative, a series of planned astrobiological missions to Venus. Janusz, the floor is yours.
Thank you very much for this kind introduction and for the invitation to give this lecture. I mean, I hope that you will find all this research that we do quite interesting. And I know that at the first glance, it might actually sound like science fiction, but I hope that by the end of this talk, you are going to realize that indeed, quite possibly, water is not the only solvent for life. And well, this is something that we actually now study quite intensively, and we have to realize that yes, of course, water is actually quite common solvent. Liquid water is quite common solvent in the solar system. I mean, we all know that there is a liquid water ocean under the surface of Jupiter's moon Europa, for example, or maybe also Enceladus in the Saturnian system. But there are other liquids in the solar system as well on other worlds, either present or suspected to be present. And of course, for example, we have Venus, we have Io, the Jupiter's moon Io, and then of course Titan, the Saturn's moon, and also Triton, the moon of Neptune. And each of those example bodies in our own solar system have some sort of liquids, or at least they are suspected to have. On Venus, of course, we have concentrated sulfuric acid, that is the dominant liquid, not on the surface, of course, but in the clouds. So as far as we understand the clouds of Venus, they are made of liquid droplets of concentrated sulfuric acid. On Io, people are speculating that maybe under the surface of Io, the most volcanically active body in our own solar system, there might be actually reservoirs of liquid sulfur. And if any of you actually ever seen liquid sulfur, now, well, this is actually a pretty beautiful solvent. It's blood red, and it actually is present also on Earth, rarely, but it is. Sometimes if the crater of the volcano is hot or still warm after after the eruption, the sulfur deposits often melt and create this very, very alien-looking sort of sulfur-based ecosystem or biome. Not biome in a sense. I mean, this environment in which this liquid sulfur actually dominates and creates these temporary streams or lakes or little reservoirs of sulfur that eventually solidify. So such bodies potentially could also exist under the surface of Io, maybe even on some very alien-looking exoplanets. If we actually move to the outer solar system even further, we have, of course, Titan, which is the moon of Saturn, as I mentioned, and this is the only place in our solar system, apart from Earth, that has liquid reservoirs on the surface. Yes. So, but in this case, of course, these are very, very cold substances. These are so-called cryosolvents, and in this case, this is liquid ethane and methane. And if we go even further in our solar system, we can actually visit a Triton, which is essentially the moon of Neptune. And people were speculating, at least since the 90s, that liquid nitrogen could play a role on this moon as well. For example, it could exist in the form of temporary geysers. This is not yet confirmed, but nevertheless, people also speculate that this could be possible. So we have all kinds of different liquids that shape very many different bodies, and in our own solar system, and also many different chemistries could actually happen in these very alien conditions. So this is part of our research as well that we do. We actually try to answer questions on basically what type of chemistry and which of these solvents actually could potentially be alternatives for water for life, essentially. And before we actually answer the question which of the solvents are actually good for life, we have to actually define what liquid actually could be a good solvent for life. And we actually spent quite a while thinking about this problem, and we boiled down this question to essentially 4 different categories that each solvent actually should have to be a good solvent for life. And first of all, these solvents, they have to be common. So there is a criterion of occurrence. So the solvent that is a candidate solvent for life or for some complex organic chemistry has to be stable as a liquid on the planetary surface or in the crust or in the clouds. Essentially, this cannot be some sort of obscure laboratory solvent, for example, or some solvent that cannot form in sufficient quantities on the in the planetary setting. We, of course, have a criterion of solvation. And this is quite important criterion because the solvent has to be able to actually actively dissolve various different compounds, but it cannot dissolve everything. And this is quite important because if we actually have a solvent that dissolves everything, then various, for example, cellular structures or membranes like cell walls wouldn't be able to form. Some molecules so there has to be this balance between dissolving various different compounds, even very large polymers, for example, like genetic polymers or various enzymes or proteins and so on, and the insolubility of other components of life, like, for example, components that could build cell walls or cell membranes. And of course, we also have the requirements for solute stability. And what we mean by that is that if the solvent actually comes into contact with its various molecules, either dissolved or insoluble, they have to be stable in the presence of this solvent. So if we have a solvent that is very chemically reactive, for example, and destroys these various other chemicals rather readily, then such solvent cannot be a good candidate for a solvent for life, for obvious reasons. And finally, we have the 4th criterion, which is the so-called solvent chemical functionality. And what we mean by that is that the solvent has to actively participate in metabolism or in our cellular physiology or in biochemistry, and not just be a passive support for our biochemistry. This might not be an absolute requirement, because we could, in principle, imagine a situation in which the solvent just dissolves certain molecules and brings them together so they can act in biochemistry, or they can form metabolism. But and essentially it can just provide the passive support without actually participating in it. But we decided that to include it here because obviously water is such solvent that is actively participating in our biochemistry. For example, we have various hydrolysis reactions, like for example breaking peptides apart, or breaking various nucleic acids apart. This is all reactivity that is dependent on water and its active role in our biochemistry. So we also consider this to be an important part. So we recently actually did a survey of all of the solvents that were postulated as potential candidates for solvents for life, or some substitutes for life. This paper is currently actually published like maybe 2 days ago, so you are welcome to see it for yourself. But and obviously from this work, we can clearly see that there are certain solvents that stand out. Water obviously works. It meets all of these 4 criteria that I've mentioned on the previous slide. But there is another solvent that is actually quite interesting, that meets all of the criteria, with maybe a little bit of questions when it comes to solute stability. And this solvent is concentrated sulfuric acid. So concentrated sulfuric acid is actually quite common. We know that on the sort of galactic planetary scale. It dissolves the molecules quite nicely, but it doesn't dissolve everything. And essentially, of course, also has the chemical functionality. And the only question that we actually do not really exactly know, because people rarely study this, is how stable molecules or complex organic molecules are in this solvent. Because we know that concentrated sulfuric acid, at least from our point of view, appears to be a very, very aggressive solvent. So we, of course, are interested in concentrated sulfuric acid because of Venus, our planetary neighbor. It is, as I mentioned, the place where concentrated sulfuric acid is the dominant liquid. And before we actually dive into the sulfuric acid clouds of Venus, we have to remind ourselves what Venus actually is and what are its characteristics. It is often called sister planet to Earth, and this is actually quite misleading, because the conditions on Venus are actually very, very different than what we have here on Earth. And especially, for example, the temperature on the surface of the planet is very, very high. It reaches approximately 465 Celsius degrees, which this actually makes this planet the hottest planet in our own solar system. The surface pressure is also quite high. It's more than 90 times higher than what we have here on Earth. But what really makes this planet to stand out is the atmosphere. So the atmosphere, where the dominant gas is actually carbon dioxide, and the clouds. This is what actually makes Venus quite special. What we have there is basically the cloud decks, or clouds that are continuous and permanent. What it means is essentially that they cover the entire planet with a blanket of clouds. So this is not like we have clouds here on Earth made from liquid water droplets that can form and dissipate within, let's say, 1 hour time. There on Venus, the clouds are actually perpetual. They are permanent, and they are continuous. And what is even more important is that we also call them temperate. And what we mean by that is that at the altitudes where they exist, around, let's say, 50 kilometers above the surface, the temperatures that we encounter there are quite benign. They are from, let's say, 100 to 80 degrees in the bottom clouds to, let's say, 0 to minus 20 Celsius degrees at the top of the cloud. So these are the ranges of temperatures that are actually quite conducive in principle, of course, to the complex organic chemistry, or who knows, hypothetically, even life. But of course, the devil is always in the details, because we have to, again, remind ourselves that these clouds have nothing to do really with our own clouds here on Earth. There, the clouds are made of liquid concentrated sulfuric acid. So not really water. And this environmental this is actually an environmental challenge that is unlike any challenge for life that life has to endure here on Earth. Because we have to realize that the concentrated sulfuric acid environment, at least from our point of view, of our own life, is actually many more times more acidic than even the most acidic places that we see here on Earth. So even these very already alien-looking Dalol pools in northern Afar in Ethiopia, which are basically like quite acidic conditions, are still, water pools that have just high concentration of acid, but it is still a diluted acid. It's nothing like the acidity of the clouds of Venus. And finally, we also have very low water activity. The clouds of Venus, as far as we understand them, of course, are 50 to 100 times more drier than the Atacama Desert, which is one of the driest places on Earth. So if we actually envision life that exists in the clouds of Venus, and it's still somehow based on water, then it has to have evolutionary adaptations and some evolutionary solutions that have no precedent here on Earth. And this essentially brings us to the very, very important conclusion when it comes to life in the Venusian clouds, is that if Venusian life exists in the clouds, and this is of course quite hypothetical, then it has to be life as we don't know it. It has to be life that is very, very different than life that we have here on Earth, because of these two challenges that actually make our life virtually impossible in the clouds of Venus. So what we actually try to ask ourselves, can we actually embrace this concentrated sulfuric acid? We do not consider these two challenges that I've mentioned on my previous slide as an absolute challenge. We actually want this to be part of the environment in which life could exist. And can we actually use the concentrated sulfuric acid as a solvent for life instead of water? Yes, because the clouds of Venus have liquid droplets there. This life needs liquid. The liquid actually can dissolve various different molecules, bring them together so they can form various chemical, or so they can react and create potentially complex chemical networks or reactivity of molecules. So can we actually have this sulfuric acid to act as a solvent for life rather than water? And at the first glance, this might sound like a completely mad, ridiculous question, because if you actually Googled or if you searched on YouTube for sulfuric acid and sugar, for example, then you will quickly find many different videos that show what happens when you add concentrated sulfuric acid to various components, various, for example, chemicals that are common in our own biochemistry, like, for example, sugars. And what will happen is essentially, after less than a minute, you would create this very unwelcoming looking black blob of carbonaceous mass that has nothing to do whatsoever with complex organic chemistry, or even not to mention life. But what is actually quite important is that this reaction or these movies are actually quite misleading, because this doesn't mean that organic chemistry itself is incompatible with concentrated sulfuric acid. It just means that sugars are incompatible with concentrated sulfuric acid. This is we study, and we currently, one of our main interests is essentially studying the organic chemistry in concentrated sulfuric acid. And by we, I mean our group as well as our collaborators, like, for example, Professor Steven Benner and Dr. Janusz Pętkowski. And they, for example, were able to show that there is formation of various different, there are certain interesting chemical reactions, like, for example, the formation of new carbon-carbon bonds in concentrated sulfuric acid. And this, in turn, leads to the formation of various different interesting polymers and reactivity that is actually very, very specific to this aggressive solvent. And what is quite even more important is that this type of reactivity happens, or this formation of complex organic chemistry can happen from very, very simple components, from, for example, atmospheric gases like, for example, formaldehyde or carbon monoxide. So those are components that could in principle exist in the clouds of Venus or do exist, like, for example, the carbon monoxide gas. We actually asked even more basic question, which of the building blocks of our own biochemistry are really unstable in concentrated sulfuric acid, because nobody actually did such a comprehensive survey of stability of our life's building blocks. We know sugars, of course, are completely unstable, but what about, for example, amino acids? And this is a wonderful work of Max Ziegler, where he was able actually to show that 19 out of 20 of the amino acids that build our proteins are actually quite stable in concentrated sulfuric acid. Sometimes they undergo some chemical modifications, like, for example, sulfations and sulfonations, but this does not lead to their instability. This only shows that they can undergo some chemical modifications that in some cases are actually even quite reversible. And this only also underscores the fact that sulfuric acid also has this chemical functionality, as I mentioned at the beginning of my talk. This type of, at the first glance, very simple experiments actually are actually so unexpected that we actually got a cover of Astrobiology journal. And we decided, so after this initial quite unexpected success of our venues into organic chemistry in concentrated sulfuric acid, we decided to ask even more outrageous question, which is, what will happen to the peptide bond in concentrated sulfuric acid? And we could argue that the peptide bond that you can see here on the screen is one of the most important bonds in our own biochemistry. It is the bond that brings together amino acids in proteins and peptides. So it allows for the formation of long polymers that have this sort of executive power in our own biochemistry. For example, creating enzymes and various complex polypeptides. And at the first glance, you might think that this bond would be completely unstable in concentrated sulfuric acid. This what many biochemistry textbooks would tell you. But what we actually were able to show, and this was absolutely quite unbelievably surprising for us, is that some peptides, and here exemplified by two examples, two simple dipeptides, are actually completely stable in concentrated sulfuric acid for many, many months. So, for example, here you have examples of 4 different dipeptides: alanine-alanine, glycine-glycine, glycine-alanine, and alanine-glycine. And we can see that some of them are actually quite reactive. They react and form some complex products, like, for example, which is exemplified, which basically is visible here by these amber-looking solutions. But some other peptides are actually completely stable and remain dissolved and remain soluble for many, many months in concentrated sulfuric acid. So this is actually a good moment to remind ourselves that there is a very big difference between diluted acid and concentrated sulfuric acid. So these two substances are actually very different and have very different chemical characteristics. In diluted acids, all dipeptides, and in our case, or our model dipeptides in a sense, undergo canonical acid catalyzed hydrolysis. So this is what normally you would think happens. And this peptide bond that is marked here in red essentially breaks apart. And when it breaks apart, you release the individual amino acids that build this dipeptide. So in this case, what happens is water is the dominant reactive species that actually is responsible for the breakage of this bond and release and for the breakage of this alanine-alanine dipeptide. But in concentrated sulfuric acid, a different situation takes place. In concentrated sulfuric acid, water actually does not dominate this reaction mixture. And what happens is that you have some dipeptides that can be completely stable. And when they do react, the dominant products are actually not the original amino acids that build the dipeptide, in this case, the alanine-alanine dipeptide. but something else. We have we different bond in this case breaks and splits this molecule apart, and the dominant product is actually not an individual amino acid alanine, but rather a modified version of amino acid alanine, which is called alanine amide, so an amide variant of alanine, and a reactive product that is called an acrylic acid. And acrylic acid is actually a very reactive molecule that then polymerizes and reacts further in concentrated sulfuric acid and into complex products. And this is what gives these solutions on the previous slide this sort of amber-looking color. So what happens here is essentially what is key for this red bond to break is the fact that you have these carbon hydrogen bonds on the on the side chain of the alanine. Without that, this reaction, this is a key to formation of this double bond and breakage of this red bond between the alpha carbon and the nitrogen. So as you can see, you can have completely different reactivity depending on the concentration, on whether your acid is actually diluted or concentrated. So what we actually asked ourselves is that if we have a mechanism or if we understand correctly how this reaction actually proceeds and what functional groups are important for this dipeptide, the alanine dipeptide to break, then we can actually then try to stabilize peptides or dipeptides in this case in concentrated sulfuric acid indefinitely. So what we actually did is we created a variant of this alanine dipeptide, where all these reactive carbon-hydrogen bonds on the side chain of alanine were substituted with fluorine. And this, of course, prevented this, our hypothesis is that this would prevent this type of reactivity and stabilize the dipeptide indefinitely in concentrated sulfuric acid, or at least for many, many months as we measured the reaction. And indeed, this is what happened when we have a situation in which you can dissolve these dipeptides in concentrated sulfuric acid, and you can monitor their stability with NMR spectroscopy. And what you can clearly see here is that the solutions for this modified fluorinated dipeptides are actually remain clear and unreacted for many, many months. So what is the moral of this story, or a conclusion of this story, is that you shouldn't assume anything before you actually do an experiment, because this type of stability and behavior of the peptides in concentrated sulfuric acid would be very, very difficult to predict. And nobody actually expected this stability to happen, and yet it did. So as you can see, the concentrated sulfuric acid is not necessarily hostile to peptides. In parallel, we asked ourselves, of course, a different question: what about the genetic part of our biochemistry? We know that the executive part of our biochemistry could, in principle, be stabilized if modified. What about for example, nucleic acid bases? The famous guanine, cytosine, adenine, and thymine, and others as well. Are they going to be stable, and for how long? And again, we can say that they are very stable, that they are stable for at least a year, where we stopped measuring after a year. And the NMR spectra, in this case, are actually remain unchanged for many, many months. And of course, there are some modifications that happen. The protonation of carbonyl groups and amino groups, of course, happens as it is expected in acid. But the rings themselves, the ring structures of these molecules remain completely unchanged. And of course, there is a rhetorical question that one could ask themselves: what about the actual genetic-like polymer? Can we actually build a genetic-like polymer in concentrated sulfuric acid that could, in principle, be stable and stably dissolved in this solvent? And of course, we can remind ourselves that, yes, the YouTube videos which show the spectacular reaction of concentrated sulfuric acid with sugars are correct, because, of course, DNA cannot function. DNA, RNA, these type of molecules will not be able to be, will not be stable in concentrated sulfuric acid. They both have contained the ribose sugar, which will be very, very reactive, as well as the phosphate ester groups that are also quite reactive in concentrated sulfuric acid. So we need some sort of other type of polymer that could potentially be stable. And what we actually tried is to try to see if the other polymers, like genetic-like polymers, could in principle be stable. And we tested PNA, which is a peptide nucleic acid. And to our absolute amazement, this type of polymer is actually, well, we can stay metastable at room temperature in 98% concentrated sulfuric acid. And what is even more remarkable is that if you subscribe to the idea of a polyelectrolyte theory of the gene, you know that the backbone of the genetic polymer, no matter what the genetic polymer is, has to be charged to dominate essentially the chemical and physical properties of the entire polymer. And in DNA, the phosphate groups give the negative charge, the permanent negative charge to the polymer. But in concentrated sulfuric acid, in PNA, in a single strand of PNA in this case, the backbone, or the carbonyl groups of the backbone, are permanently and stably positively charged because of the protonation of these carbonyl groups. So you are having exactly the same sort of physics behind the polymer, in a sense that you get the stable positive charge or stable charge, but it is an opposite charge that we have here in water. And you might say, okay, this is amazing, and we are almost there. We almost have the genetic polymer present in the concentrated sulfuric acid. Unfortunately, we are almost there, because in higher temperatures, like for example above 50 Celsius degrees, this PNA polymer becomes unstable. It breaks, but it doesn't break like to create a sort of carbonaceous goo that doesn't have any structure to it. There is one particular bond that breaks in hot concentrated sulfuric acid, and this is this one marked in red. So we are now currently very actively work on stabilization of this bond and synthesis of polymers that actually could withstand even hot concentrated sulfuric acid as well, and essentially to stabilize this structure even further. We, of course, not yet have the answer on the formation of the double helix-like structure. We hope to answer that question at some point as well. We could, at the first glance, argue that the double helix would not form because of the protonation of amine groups and the protonation of carbonyl groups in concentrated sulfuric acid. But as I mentioned before, without actually doing an experiment on it, I would not assume anything, because 2 years ago, when we started all of this work, we assumed that no organic chemistry can actually survive in concentrated sulfuric acid, that nothing interesting can be stable there, and so on. 2 years later, we have potentially stable peptides and are even on the road to genetic-like polymers. So we are not there yet, but we might actually be quite close to the formation of a sort of genetic-like double helix that it would be stable in this aggressive solvent. And moving forward in our race through our own biochemistry, obviously we know that one of the most important points of any type of life is having a barrier, having a cell wall or membrane that would essentially demarcate what is inside and what is outside of the cell, what is mine, what is foreign. So essentially, we asked ourselves a question: can we actually have acid-resistant membranes? And this is a wonderful collaboration that we forged with a group of Professor Jack Szostak, and specifically with my friend Dr. Daniel Duzdevich and Colin Nisler from their group. And what they were able to show is that you can actually form both micelles and liposomes in concentrated sulfuric acid, and you form this rather readily. Of course, what is very important is that you cannot use our own lipids. You cannot use our own building blocks of our own membranes here on our own water biochemistry. You have to tailor your chemical Lego building blocks to this solvent, so you have a different type of lipids, but the physics behind the formation of this type of structures, like these vesicles, remains or stays the same. And I could go through all of the biophysics of the formation of these structures and explain all of these graphs to you. But as they say, as people say, seeing is believing. And this is essentially a wonderful photo that from fluorescent microscopy that Colin Nisler took. And probably if I didn't tell you that this is that these are vesicles in concentrated sulfuric acid, you would think that this is just a formation of regular vesicles in water, because they essentially look exactly the same, like they are in water. This is the particular image of vesicles in 80% sulfuric acid. This is just, I took this because this is a particularly beautiful frame that Colin took, but other concentrations of concentrated sulfuric acid also work. So this is not, there are other concentrations of sulfuric acid that work equally well. What is quite also remarkable is that you can do fluorescent microscopy in concentrated sulfuric acid, because apparently the dyes that are used for let's say, staining the membranes that intercalate into the membrane and can stain the membrane actually also are stable in concentrated sulfuric acid, so allow you to use this technique to visualize the vesicle formation. What is also quite remarkable, and this is probably even more remarkable than the formation of the vesicles themselves, is what you have here on the right side of the screen, which is the so-called perling behavior. So this is something that sometimes we see the formation of tubes and other complex structures in concentrated sulfuric acid, and these tubes sometimes tend to pearl or fragment into smaller structures, into smaller vesicles in concentrated sulfuric acid. It is very important to note that this type of behavior is not readily visible in water. It requires special conditions like special lipid composition, for example, or turbulent water flow to facilitate this type of pearling behavior. In concentrated sulfuric acid, we see this behavior readily. So this we could speculate that this actually makes the concentrated sulfuric acid actually quite conducive for the so-called maybe prebiotic vesicle division. So in summary, what I would like you to actually take home is that our group and our collaborators, we are building a new paradigm for sulfuric acid as a solvent or a potential solvent for life. We now know, thanks to our work of our collaborators like Professor Stephen Banner and Dr. Janusz Pętkowski, that rich organic chemistry in concentrated sulfuric acid is actually a fact, and it can form from very simple or more organic molecules, or even gaseous components of the atmosphere, like for example carbon monoxide gas. And this actually suggests that the presence of organic chemistry in the clouds of Venus is virtual certainty. It's just a question what the organic chemistry is, and is this alive or complex enough to essentially be considered prebiotic building blocks. We were also able to show that the nucleic acid bases of variety of nucleic acid bases, I would say, are actually also completely stable, and we can even build, or we are inching towards building a DNA-like molecules or some sort of genetic-like polymer to actually show this as an example. We can also, and this is in itself quite unexpected, but we can also stabilize the peptides. And first of all, amino acids are stable, or 19 out of 20 of them are stable. And we can also stabilize the peptides themselves by some chemical modifications. And finally, we can also form complex or sophisticated membranes, micelles, and vesicles in concentrated sulfuric acid, thanks to the wonderful work of Professor Jack Szostak, Dr. Daniel Duzdevich, and Colin Nisler. So all of this opens a completely new field of study of this organic chemistry in concentrated sulfuric acid, and maybe, who knows, even at least prebiotic chemistry in concentrated sulfuric acid, if not biochemistry. And why do we do this, actually? Well, we do this because we want to go to Venus, yes, and we are not afraid. We are not afraid of this. We want to go to Venus, and we would like to actually have a series of astrobiology-focused missions. We are not afraid of actually looking for having a bold scientific objectives and look for specifically complex or increasingly complex organic molecules in the clouds, because this the question of whether there is organic chemistry in the clouds of Venus was never asked by any mission to Venus. So none of them were actually able to detect organic chemistry because nobody asked. And we hope to actually change that. We hope to actually look for organic chemistry, and our venture in the clouds of Venus starts with the planned rocket lab mission to Venus, which will, among other objectives, will look for fluorescence of cloud particles, and this can be essentially a proxy for organic chemistry in the clouds. And then building on that, the further missions are also actually planned, or we are now actually working on developing them as well, which is to look for increasingly more complex organic molecules, or essentially elucidate this composition of the cloud particles to a greater extent. And I will end the talk with, of course, the thanks, and not only thanks to all the people that are involved in the development of the missions, but also a special thanks to all of the people that work on this wonderful organic chemistry that I showed you today, especially to Professor Sarah Ziger. Without her and her involvement, nothing that I showed you today would be possible. So huge thanks to her, and of course to the rest of the team: Max Ziger, who led the amino acid work, William Baines, John Grimes, who is our NMR expert, and also wonderful work of Daniel Duzdevich, Colin Nisrer from Jack Szostak Group, and of course also huge thanks to Professor Stephen Benner and Dr. Jan Szpaczek. Thank you very much, and I welcome any comments or any questions.
Thank you, Janusz. I had to even ask, what should I say after your lecture? Our head of the conference suggested that I'm speechless, and probably this is the correct thing, what I would like to say. I wanted to ask some question, but unfortunately, you already answered my question in the meantime.
You can ask it again. I mean, feel free to ask the question
no,
again.
Maybe
I will be able
was
to.
Thinking about the stability of the peptide bond, and you already solved the problem. I'd be interested about the shape of the proteins when you have a longer peptides in concentrated sulfuric acid. How it looks like, and
a very good question. I can maybe just tease a little bit on that topic. That is obviously a question. We are our research is sort of a baby steps towards more complex polymers or more complex chemistry. We have to realize that 2 years ago we were at the stage of adding sugar to sulfuric acid and thinking that organic chemistry in concentrated sulfuric acid is impossible. And now 2 years later, we actually think that you can actually build a complex polymers and all of that. So the obvious question is are those polymers, even if we are able to stabilize them, are those polymers fold? Do they fold properly in concentrated sulfuric acid? The answer to that question is I don't know. And it's not I don't know, it cannot be possible. It's we simply do not yet know. We need more research on that. We need to actually find also good techniques, analytical techniques that would actually help us solve this problem. Sulfuric acid is notoriously aggressive to various different analytical methods, which means that, for example, you cannot really use easily mass spec to study your stability, for example, of your various compounds, because the machinery would not really survive this concentrated sulfuric acid readily. But this is something that we work on actively. There is obviously a question whether these proteins fold. At the first glance, you would say, oh no, they would not be able to fold because sulfuric acid would actually break the delicate the hydrogen bond network that helps the polymer fold. But we have to realize that, for example, hydrophobic interactions are not that perturbed in this solvent, so maybe they actually play a bigger role in this type of behavior. We don't know. I also think that maybe there is a lot of modeling work that could actually be applied here after you parameterize, I don't know how to say this, the solvent itself, because we cannot treat it as water. We have to actually treat it as sulfuric acid, and we have to actually properly set up our initial conditions. So that's my answer. This is obviously something that we work on. We try to answer this question, but we have to first find good methods to really answer this.
Yes, and this is why I think your research is very interesting, because you have to develop new methods also to study those phenomena. So as a follow-up of my question, to go to the big picture, I would like to ask you the first question, what was asked about the entropy, and what about the self-organization?
Yeah, that's a very good question. Absolutely. So we can see that there is some self-organization. This is actually related to folding also. Yes, it's also related, for example, to folding of various proteins or any other polymers. Well, I think that paradoxically, what I think, and this is my opinion, and who knows, maybe it will change after more research is done on this. But I think that sulfuric acid, in terms of that, promoting self-organization, that sulfuric acid might actually be equally good as water is. And we sort of see this from for example, the wonderful work of Daniel Duzdevich on the vesicles. Yes, we not only are able to see the self-organization of these vesicles, but we see complex behavior of these vesicles also, like this purling behavior. We can also, and then our paper on the vesicles is available on arXiv freely to read. We can also see that the vesicles, for example, are retaining their structure if you move them from one solvent to another. Yes, so you can move them from one solvent to another. So I think that, and the question is whether there is this self-organization happens to other type of chemistry, to polymers. I do think so, but this is work of the future, obviously, for us.
Thank you so much. So next question will be also about chemistry.
Yes, I see the question on the screen. I don't know if the viewers see this question as well. So yes, should I read the question, or is
no? I think people can see the.
Okay. So the question, in very briefly, the question is about the ammonia water eutectic as a possible solvent for life. And yes, that's actually a very interesting question in itself. So we have an entire, and I again can advertise our work that is also present on arXiv. We 2 days ago, our paper in astrobiology on the alternative solvents and these 4 criteria and all of the solvents that I showed was published. So we address this question directly, or the question of ammonia and ammonia water mixtures directly in this paper. What I can only say very briefly, because otherwise, we could sit here for 2 hours, is that ammonia has one problem. Ammonia, in contrast to other solvents that like water, for example, or like for example, the sulfuric acid as well, is actually quite unstable photochemically. So you can easily destroy ammonia by solar radiation. And the problem is that, in contrast, for example, to water, which to a degree can, for example, reform and so on after such photochemical destruction, ammonia tends to be destroyed and form nitrogen N2. And nitrogen N2 in this case is a very, very stable atmospheric component, an atmospheric gas. So once the ammonia is lost, it's actually, there is no way of reforming it and replenishing your solvent. So any life that loses its solvent reservoir on a planet and is incapable of replenishing it would likely perish. And also maybe from that point of view, this type of ammonia, sort of eutectics or ammonia as a solvent in general, is less likely to be present. So in the planetary setting, because it might essentially get destroyed. Now, this is when it comes to the surface, yes? Now, there is another chemical quirk of ammonia-water mixtures, is that essentially ammonia-water mixture is more like basic water than ammonia. Yes, so you would be closer to behavior or chemical behavior and physical behavior of water if you had an ammonia-water mixture than pure ammonia. But I highly recommend the viewers to look at our paper on this very topic, and I will probably end here because we could dive deeper into these questions.
So next question is more about the Venusian life then.
Well, hypothetical, but maybe less. But maybe less and less hypothetical with every passing NMR measurement. So, yes, if we assume. Yeah, that's an interesting question. Yes, because obviously, what we do. Okay, so there is quite a lot to unpack here in this question. So it is obvious that, let's say that we do not find native Venusian life, that there is no life in the clouds of Venus whatsoever, and that yet this organic chemistry could be there or whatever. There is nothing of that sort. Could this research of this very basic chemistry in concentrated sulfuric acid be actually useful for something? Well, yes, we don't know to what it was going to be useful immediately, but maybe this is also part of synthetic chemistry, synthetic biology also, and it's at least a synthetic biology in its very in its infancy, because who knows? Maybe we would, if we actually inch towards much more complex structures, maybe we will be able to actually create some sort of eventually. I don't say that this is going to be anytime soon. Some very very different quasi-like lifelike structures that are going to use this solvent as a main liquid. Now, there is an obvious application, I should say, quotation mark, but obvious relevance to maybe not really Venus per se, only to Venus, but also exoplanets that contain sulfuric acid. We are quite confident that sulfuric acid is actually a very common substance in the galaxy. So the planets like Venus, or even planets where sulfuric acid could be some solvent on the surface of the planet, if there is, let's say, a little bit colder Venus that allows sulfuric acid to actually reach the surface and persist on the surface, might actually be present. And in this case, this type of research also tells us what type of organic chemistry could we actually expect on exoplanets. Of course, this is a bit of a theoretical question because as this for now, we will not be able to verify it with any meaningful exoplanetary data. But yeah, a very, very cool question. I guess we will know when we will know where we do not really know where the future will bring us, what future will bring us, but we work on it very hard. Yeah, very good question. It's the question about what would be the path to origin of life in concentrated sulfuric acid. Well, I will say the following. We do not even know how life originated on Earth in presumably water, although we do not even know that. So asking questions, of course, those are valid questions of are there any paths to make this prebiotic or non-biotic, abiotic chemistry, organic chemistry in concentrated sulfuric acid into something alive, well, are a bit far-fetched for now. We don't know. But what is quite surprising and what is actually maybe giving us a bit hope is that the same physics, for example, for the vesicle formation, applies to both solvents. That from work of the Dr. Janusz Pętkowski and Professor Stephen Benner, we know that this sort of complex polymerization reactions do happen. So it's not that this solvent is actually preventing this complex organic chemistry from arising. Now the question is, can we actually have some sort of selection or some evolution to start acting on these complex mixtures so they can, stop being just chemistry and become biology. But as I mentioned, I mean, we do not even know how this happened in water. So the question how this happened in concentrated sulfuric acid is well, it's much more difficult. But I will finish with the following. It is 100-year anniversary of the Oparin's book on the origin of life. Yes, today it was published in 1924. So we have 100 years year anniversary of this book, and we still didn't figure out how life originated in water. Our work on organic chemistry in concentrated sulfuric acid is 2 years old. So we still have some catching up to do, and hopefully we will be able to show something more substantial in the origin of life field in this solvent as well. Yes, a question. Yeah, this is another cool question. Thank you very much. The question is, what do we know about the species that can survive in low or sub-inhibitory concentrations of sulfuric acid? And is that within the scope of my research, or our research, I should say? I would say that this is definitely something that should be studied. So there is a persistent idea in the extremophile, for example, community of some that there are some limits to life on Earth. That, for example, water activity has a limit, and so on, that below which life cannot exist or cannot survive. And this probably also applies to acid. Now, I would be very careful. So now this is so that's why it is actually a perfect question, because somebody should actually try to do a long-term evolutionary experiment. Yes, increase these stresses. From our point of view, it is a stress. From the point of view of hypothetical Venusians, it's actually an everyday life. So it's not an extreme environment at all. But somebody should try to increase these stresses very gradually and see how does this affect the organisms how do they deal with this environment, and do they survive? There is one very important point that we have to make. It is possible that our life cannot adapt to, for example, low water activity or maybe even high acidity, because there is no such conditions on Earth, and our planet is always wet, even if it is very dry, like Atacama Desert. Sometimes Atacama Desert gets completely flooded. So any extremophilic organism has to be adapted to both conditions, has to be adapted to high water conditions as well as the low water activity conditions. This might actually be a problem, and this is where the extreme might come in, the adaptations to both extremes of both worlds. Venusian clouds are perpetually and permanently dry and are actually an extremely stable environment. So in other words, flooding is not something that a Venusian organism, even if it is, let's say that it is based on water, and it is more similar to our own life than the very hypothetical sulfuric acid-based life, it never has to adapt to both flooding conditions, or so high water activity conditions, and low water activity conditions at the same time. It might actually be easier for life in general to adapt to one extreme, knowing that the environment in which you live is actually very stable. So I leave it at that.
Thank you so much. Next question will be shown in 2 parts, or? Yes.
Yes. Most of the relevant long-range interactions. Oh, sorry, I didn't see the.
No, and this is the second question. This is the follow-up to the first entropical question. The first question is about this analogous reactions for the water.
Oh yeah. So the question is, the broad question is about the protonation. In fact, yes, absolutely. In concentrated sulfuric acid, many groups are going to be protonated. The amine groups, the carbonyl groups, and so on and so on. Now, and by the way, concentrated sulfuric acid does not have pH 1. It actually has the pH scale. It goes way beyond the pH scale into Hammett acidity of -12 or so, or something like that. So it's even more. So the situation is even more grim from that point of view. But I would but going back to the protonation, yes, of course you have to assume that the protonation is going to be there, and it will affect how the molecules behave. It will affect how these molecules interact with each other. It will affect how, for example, the metals are complexed. Who knows? I mean, how they can be dissolved in the concentrated sulfuric acid, and can they be actually captured by these polymers? This goes back to the same questions that we had at the very beginning: how these polymers, how these various organic chemistry actually folds in this concentrated sulfuric acid. And for now, we don't know. And we don't know, for example, what is even sometimes the protonation, the exact protonation state of all the groups in these polymers, for example. So there is a lot of work for the future here on the sort of chemical details that are essentially very specific to each of these specific molecules that I talked about. Yeah, these are fantastic questions, because these are all questions. I didn't see the question entirely, but these are are they and can they be an analogous reactions to let's say the polymerization, so the condensation and also sort of the hydrolysis of these polymers that Earth's life chemistry uses, and where water actually has an active role. So that's why water has this sort of important chemical functionality here. Maybe. I mean, this dipeptide reactivity, to a degree, is example that this sulfuric acid indeed participates in this sort of breaking of these molecules, and it breaks them in a specific manner. So it's not like some chaos in this test tube. It's actually something but how this can happen at this stage, I don't know. I could also speculate that we have to remember that sulfuric acid, even the very concentrated one, is never alone. It always has this water in it. So maybe there is also presence. There is also place for this water to play an more active role in the fully fleshed out concentrated sulfuric acid biochemistry. But of course, that would be a speculation. At this stage, we do not have an example of such condensation, breakage and condensation pair for this type of reactions that we have in biochemistry. But this doesn't mean that such reactions do not happen or are impossible. It means that we just didn't have time yet to look into such details.
Okay, that was the last question. Thank you one more time, Janusz, for your lecture and for the Q&A session. I think that was a perfect start for our conference. So also quite a follow-up of your lecture. Next session, synthetic and gravitational biology, part 1. Part 2 is tomorrow. Part 1 is in 15 minutes. 9 p.m. Central European time. So see you during the next session.
Thank you very much.
Thank you so much.
That was a pleasure. Yeah. Thank you. Bye.
Bye.